In this video, we're going to talk about how fungi reproduce. Now, before we get too far into fungal reproduction, let's do a quick review of what these terms haploid and diploid mean. These are terms that you probably have heard before when you've learned about mitosis and meiosis. Haploid means that an organism has one set of chromosomes. Diploid means that an organism has two sets of chromosomes. And these two sets of chromosomes, we typically call these paired chromosomes. And these paired chromosomes are also known as homologous chromosomes. The paired homologous chromosomes come from sexual reproduction. So one set comes from one organism and one set comes from a different organism. So we typically say one set comes from mom and one set comes from dad. Now with organisms like fungi, we don't really have a mom and a dad, but we have one set that comes from one fungus and another set that comes from a genetically different fungus. We typically write haploid as 1N, and diploid we refer to as 2N. Now, both haploid and diploid organisms can go through mitosis. But only diploid organisms can go through meiosis. Fungi can reproduce either asexually or asexually and sexually with an alternation of generations. And what this alternation of generation means is that at some points in the life cycle, the cells in the fungus are going to be haploid, or 1N, and at other times the cells are going to be diploid, or 2N. And alternation of generations is something that happens in humans as well. So we will compare these when we get to that particular slide. Asexual reproduction can include fragmentation, budding, or spore production, and we're going to talk about all of these in more detail on the following slides. Fragmentation occurs when fragments of the hyphae form new colonies. So remember, we've talked about the mushroom, and then the hyphae are these strings, kind of like the roots that are underground. Remember that these hyphae are made up of cells. And so what can happen is that part of this hyphae can break off, and then it can start to go through mitosis in order to form more cells, which ultimately forms a new hypha and ultimately forms a new mycelium. Budding is a form of asexual reproduction that occurs in yeast. And what happens is that mitosis occurs, but a bulge forms during cytokinesis, and then the daughter cell detaches from the parent. And you can see that happening here. So this is a microscope looking at yeast, and you can see the kind of the parent yeast cell here. And then you can see the bud coming off here. And this bud will detach and turn into its own yeast cell. Fungi can also reproduce asexually through the production of spores. And more specifically, we call these asexual spores. And that's because we're going to talk about sexual spores later on. So during this spore production, you can see that we have this mycelium. And remember, the mycelium, we can also call it the thallus. and it's made up of the hyphae. And so the cells of the mycelium can go through mitosis. Remember, mitosis is a form of asexual reproduction to produce these spores. And one thing to keep in mind with asexual reproduction is that these spores are genetically identical. And they're genetically identical to each other and also genetically identical to the original parent cell. And this is important because genetic diversity is typically considered beneficial in unstable environments, but asexual reproduction can be beneficial in stable environments. And we'll talk a little bit more about why that is the case a little bit later on. So once these asexual spores are produced by mitosis, germination can occur. And germination just means that those spores can find a suitable place to, well, they land, and hopefully they land in a suitable place where the spore can then start going through mitosis in order to start making a new mycelium, which will ultimately make a new fungal organism. Another thing to point out here is that the cells in the mycelium are haploid. You can see that it says 1N. And because we are only doing asexual reproduction here, it means that the spores are also haploid. And then they also produce haploid cells when they germinate and start forming a new mycelium. Some spores are released within a protective sac called a sporangium, and they are often transported by the wind or they hitch a ride on animals. This picture here shows a puffball fungus, and you can see that this puffball here essentially pops open and the spores are dispersed into the air where they are carried by the wind. Some fungi also reproduce sexually, and one benefit of this is that it helps to increase genetic diversity, and we'll talk about why that's important a little bit later on another slide. So we mentioned that alternation of generations, and so if the environment is changing or there are adverse environmental conditions, then a fungus that might otherwise reproduce asexually can actually switch to sexual reproduction in order to give their spores a better chance at survival. Now, with sexual reproduction, there are two genetically different mating types involved, and with fungus, we refer to those as just positive and negative. But this is similar to what we see in humans, where we have male and female. However, with fungi, we don't have the genetic kind of sex determination. So we don't have like XX or XY chromosomes, and they don't really have the kind of genitalia structures that would make one be considered male versus female. So it's not exactly like male versus female. It just means that there are two cells involved, and those cells are genetically different from each other. Now, some fungi might be homothallic, and homo means same. Remember, the thallus refers to the mycelium. And fungi that are homothallic are able to self-fertilize. And so essentially there are cells in the same mycelium that represent these two different mating types. And so just two cells from the same mycelium can fuse or merge in order to conduct sexual reproduction. Heterothallic, hetero means different. So in a heterothallic fungus, there have to be two different mycelia involved. So this would mean two different fungal organisms, each contributing a genetically different cell to this sexual reproduction. So to kind of draw this out, if we look at the homothallic first. So remember, we've got our mushroom, and then we've got our hyphae underground. And these hyphae are making up the mycelium. Now, some of the cells in this mycelium might be the plus mating type, and some might be the negative mating type. And so homothallic fungi can essentially produce cells from these two different mating types that can merge together in order to form a cell that has the different types of genetics. Now with heterothallic, we would have two mushrooms, each with their own mycelium. And one might have the positive mating type, and one might have the negative mating type. And a cell from the positive mating type would combine with a cell from the negative mating type in order to be involved in sexual reproduction. So in this image right here, we're kind of looking at the kind of a generic fungal life cycle. When we talk about the specific phyla of fungi, we'll talk about some more specifics as to how the different types of fungi reproduce. But all of them follow these same kind of basic 4 steps with just some additional specifics thrown in there. Now, earlier when we talked about the asexual production of spores, we were talking about this asexual reproduction here. So we've already seen kind of the top part of this diagram a couple slides back. And so remember, we were taking this haploid mycelium, going through mitosis to form haploid spores that are genetically identical. And they're genetically identical to each other and to the original mycelium. And then those cells can germinate if they find a suitable environment where they can then form a new mycelium. Now, the bottom half of this graphic is where we talk about sexual reproduction. And remember, some fungi are going to do the asexual reproduction over and over. And then every once in a while, they might switch and start doing the sexual reproduction depending on environmental conditions. So the 4 stages of sexual reproduction, the first one is plasmagamy, which we can see right here. The second step is karyogamy. The third step is meiosis. And the fourth step is germination. And we're going to break these down in more detail on the following slides. So we're going to take a look at this first stage, plasmagamy, first. And this word plasmagamy can be translated into cytoplasm marriage. So plasmo, think of the plasma membrane. So 2 plasma membranes of cells are going to join together. And so essentially you're going to have cell number 1. And remember, this is going to be a haploid nucleus. And then you're going to have a second cell, also with a haploid nucleus, but notice that I drew them in different colors. And that's because these 2 haploid nuclei are genetically different from each other. Now, during plasmagamy, what happens is that the cell membranes are going to fuse, but the haploid nuclei are not going to fuse. So what we end up with is one cell with 2 separate haploid nuclei. And this is called a dikaryon. And di means 2, and this prefix of kary or karyo is related to nucleus. So essentially it means 2 nuclei. And remember that these nuclei are both haploid. So they only have one set of chromosomes each. This dikaryon can also be known as a heterokaryotic cell. I remember with haploid, we represent haploid as 1N, and we represent diploid as 2N. And so a dikaryon doesn't really fit either of those classifications. So we refer to this as N plus N. So there are essentially two haploid nuclei, but the nuclei have not fused to form a diploid nucleus yet. And you can see all of that happening in the picture here. And you can see that this cell that I drew here matches the one that can be seen here. Now, this condition of having this dikaryon can exist for a really long time, potentially, days, weeks, months, or even years. And so the cells might stay in this stage until environmental conditions change to a point that allows them to enter the next stage. The second stage is karyogamy. And remember that this prefix of karyo has to do with the nucleus. So this word karyogamy means nuclear marriage. So remember that in plasmagamy, we ended up producing a dikaryon. And so we had the cell that had two haploid nuclei. So the cell membrane and the cytoplasm have already been joined in plasmagamy. But now what happens is that the two nuclei are going to merge together in order to form a diploid nucleus. And remember that these nuclei are membrane bound because fungi are eukaryotes. So it's the nuclear membranes that are actually merging together, bringing the genetic material inside the nucleus together with the genetic material inside the other nucleus. And this resulting cell that forms with this diploid nucleus is known as a zygote. And you can see this on the image over here. This is our karyogamy. You can see that the picture I drew is matching the zygote in the picture. Now, one key thing about what has happened here is we have now, or the fungus has now brought together two different sets of chromosomes. And so we have now changed the genetics of the original cells. So the original cells only had half of the number of chromosomes, they only had one set each. Now both of those sets have come together. So the zygote now has twice as many chromosomes, and we have essentially mixed and matched these different genetics from these two original cells that came together and then fused their nuclei together. So if we were to kind of draw that out, so remember that eukaryotes have bar-shaped chromosomes. So the haploid nuclei would have one set of these bar-shaped chromosomes. And I'm just going to draw 3 chromosomes just for ease of drawing this here. And so inside this nucleus here, you would find this one set of chromosomes. In the red nucleus here, you would find a set of 3 chromosomes that are the same chromosomes, but maybe have slightly different genetic codes on them. So then in this zygote, what we end up with in this one nucleus here, we're going to end up with 2 sets of chromosomes that are now paired up. And 3 of these chromosomes came from the original black nucleus. And the other 3 came from the original red nucleus. And they are all found inside this diploid nucleus now. The third stage of sexual reproduction is meiosis. And in meiosis, what happens is that a diploid zygote is ultimately going to produce haploid spores. So remember, we start out with this zygote here that we made during karyogamy. And remember, inside our nucleus, we have these kind of 2 sets of chromosomes. Now, we're not going to go through all the kind of different stages, kind of intermediate stages of meiosis, but the end result is that we end up with 4 daughter cells. But these daughter cells are going to be haploid. Remember, they originally came from a diploid parent cell. And during this process of meiosis, these paired chromosomes are essentially going to be shuffled and then dealt out kind of randomly into these daughter cells. So these daughter cells might have a combination of, let's say, maybe 2 black chromosomes and a red one. And then maybe this one has 2 red chromosomes and a black one. And then maybe this one has 3 black chromosomes, and this one might have 3 red chromosomes. So these 4 haploid daughter cells are genetically different from each other. So they're genetically different from each other. They are also genetically different from the parent cell. And so this is helping to increase that genetic diversity in the population of these fungi. So these spores, so these are the sexual spores. Remember, we talked about up here with asexual reproduction, we have asexual spores. And remember, asexual spores do not have genetic diversity, whereas sexual spores do. And so this meiosis, we take this 2N zygote and we form these spores, which are haploid 1N spores here. And these spores may develop in a gametangia organ, which is essentially just part of the fungus that acts like a gonad. And you can see on this picture here that we are talking about kind of this area of the image. And so we're talking about meiosis occurring. And these daughter cells that I talked about over here are representing these sexual spores that are being produced. And then the 4th step is germination. And so the spores that were produced during meiosis are going to be released into the environment. And they're going to be carried primarily on or with the wind, like through the air, or they might hitch a ride on an animal as well. If that spore lands on a spot that has a suitable environment, then it is going to go through mitosis in order to form a new mycelium. So the spores might go through mitosis. We'll write mitosis here. So this arrow kind of represents mitosis. And you can see that, remember here, the spores here are 1N, and the mycelium is also 1N. So these haploid spores go through mitosis in order to form the haploid cells of a mycelium. Now, we mentioned that some fungi go through this process of asexual reproduction, but then they can also switch to sexual reproduction and then back to asexual. And we call this the alternation of generations. So it just means that these fungi are able to do asexual reproduction and sexual reproduction. So, when the environmental conditions are stable, usually asexual reproduction is favored. The reason why this is because it takes fewer resources and energy in order to do it. The fungus does not need to find a different mating type in order to occur because it is asexual. It's just happening through mitosis. So, if the environment is stable, we also know that the fungi in this stable environment are well suited for that environment because they're surviving. So we don't really need the genetic diversity in order for the population of fungi to be okay. However, when the environmental conditions change or become unstable, sexual reproduction is favored. And that's because we are increasing genetic diversity. Remember, that's going to happen through our karyogamy. Because remember, with karyogamy, we are taking two different haploid nuclei and we're fusing them together. And so we're making this diploid nucleus that now has genetic information from two different parent organisms. And then during meiosis, those diploid chromosomes get kind of shuffled around and then dealt into the daughter cells, which are the spores. And so the spores end up having a different combination of genetics from each other, and they're also genetically different from the parent cell. So this increased genetic diversity leads to better survival within the population. So it doesn't necessarily mean that the individual spores are going to have a better survival advantage. It means that some of them are going to survive and others are not. But overall, as a population, at least some of those spores are more likely to survive and reproduce. Now, you might be wondering, why don't they do sexual reproduction all the time? If this genetic diversity is beneficial, why not just always do it? And that's because it takes more energy and resources in order for this to occur. It also might be hard to find a mate. So remember that with sexual reproduction, especially with heterothallic fungi, you have to have these two different mating types coming from two different organisms. And those organisms have to be close enough together that their mating types or their mating cells can physically come together. All right, so let's fill in a blank diagram of fungal reproduction just to help us kind of review what we've talked about on the last several slides. So first of all, this picture in the middle here, we're looking at a mycelium. Remember, the mycelium is haploid, so we can designate that as 1N. And remember, the mycelium are just made up of each of these individual strands called hyphae. And the mycelium is also sometimes referred to as the thallus. Now, when a fungus is going through asexual reproduction, this arrow is going to represent mitosis. Remember that mitosis is a form of asexual cell division. And so what happens is that these cells in the mycelium go through mitosis and they produce these asexual spores. These asexual spores are haploid, so 1N, they have one set of chromosomes. And remember, they are genetically identical. And they are genetically identical to each other and genetically identical to the original mycelium. Now, these spores can then be released into the environment and they're going to land on different surfaces. And if they find a surface conducive to growth, they might go through the process of germination. And during germination, essentially what happens is that those spores are going to, in this kind of favorable environment, they're going to go through mitosis in order to form a new mycelium. And remember that there are benefits to asexual reproduction. So the fungi don't need to find a mate. All right, so the fungus can reproduce on its own. It doesn't need to find another fungus in order to reproduce. There are fewer resources needed compared to sexual reproduction, so less energy and resources needed. Genetic diversity also isn't needed because if this is a stable, favorable environment, then we know that the fungi alive in this environment are well suited to that environment. So the genetic diversity is not needed. The genetics that are already there are working just fine. And it might actually be genetically, or it might actually be beneficial to the population to not have genetic diversity because the genetics currently in the population might be the best genetics for that environment at that time. Now, when the fungus goes through sexual reproduction, we have some more steps that occur. So the first step is going to be the plasmagamy. And this is where you have cells from a positive and negative mating types fuse their cell membrane and cytoplasm. And what you end up forming is a dikaryon, which we can also represent as N plus N. And this is also referred to as a heterokaryotic cell. So you can see that we have two nuclei in the cell, and these are two haploid nuclei. In stage 2, we have karyogamy, in which the nuclei fuse. So you have the two haploid nuclei fuse in order to form a diploid nucleus. And this forms a cell called a zygote. And we represent the zygote as 2N because now we have two sets of chromosomes in this one nucleus instead of having two nuclei with a haploid number of chromosomes. The third step is meiosis. And in meiosis, the diploid nucleus goes through a couple of rounds of cell division and ultimately produces daughter cells with haploid nuclei. And these daughter cells are the spores. So the spores here are 1N, so they are haploid. And these spores might be produced inside a specialized structure called a gametangia. Once the spores are produced and released, germination may occur. And germination just means that the spores are going to land somewhere in the environment, and if the environmental conditions are conducive, the spores are going to go through mitosis to produce a new mycelium. So, the benefits of sexual reproduction is that we do have an increase in genetic diversity. So, this is really helpful when the environment is changing. But sexual reproduction has the downfalls of a mate or an opposite mating type needs to be involved, and it requires more energy and resources. So there are the trade offs between increased genetic diversity, which may help the population survive in a changing environment. But in order to do sexual reproduction, you have to have more energy, more resources, and more individuals in order to sexually reproduce.
So now let's do an overview of kind of everything we've talked about on all the other slides and put it together on this one slide on this one diagram. So first of all, I'm going to draw a line to divide the asexual reproduction from the sexual reproduction. So remember that this structure in the middle is a mycelium, which is part of the thallus. And each one of these little arms here is a hypha. And the cells in this structure are going to be haploid, which means that they are 1N. Now, during asexual reproduction, this arrow here stands for mitosis, which is a form of asexual cell division. Mitosis forms these asexual spores, and these spores are also going to be haploid. And that's because remember that these spores are genetically identical to each other and genetically identical to the cells in the original hypha that went through mitosis to produce them. The next arrow in this diagram stands for germination. And this occurs if a spore lands in an environment that is favorable for it to start going through mitosis to form a new mycelium. So this arrow also stands for mitosis because this single-celled spore starts making copies of itself in order to form a new mycelium. Now, if we take a look at the sexual reproduction part of the diagram, we can see the 4 different events that happen during sexual reproduction. So let's start with this first arrow here. And this arrow is representing plasmagamy. Which remember stands for cytoplasm marriage. During plasmagamy, a positive mating type cell fuses with a negative mating type cell. And this forms a dikaryon. Which can also be referred to as a heterokaryotic cell. And remember that in this dikaryon, there are two haploid nuclei present. Because the cell membrane and the cytoplasm have fused together, but the nuclei have not. And we denote this condition as N plus N, because the N's are standing for the haploid nuclei present in this cell. So you can see in this picture here, this is our dikaryon, and you can see the two nuclei present, and each nucleus has one set of chromosomes. The second arrow in sexual reproduction stands for karyogamy. And during karyogamy, remember the word means nuclear marriage. And during this process, the two haploid nuclei fuse to form a diploid nucleus. The cell that is formed during this process is known as a zygote. The third arrow in the sexual reproduction part of this diagram is indicating meiosis. And meiosis is a form of cellular division in which the diploid nucleus that was produced in the previous step is going to go through this process to produce haploid daughter cells. And these daughter cells are going to be called spores. And you can see those spores on this diagram here. And it's important to note that these spores that were produced by sexual reproduction are genetically different from each other. And genetically different from the original cell that they were produced from. The fourth arrow on this diagram stands for germination. Of the spores. So if a spore lands in an environment that is suitable for it to survive, it is going to go through mitosis to form a new mycelium. The new mycelium will then develop into an entirely new fungal organism. And recall that some fungi can switch back and forth between asexual and sexual reproduction. And this switch occurs based on environmental conditions. So asexual reproduction is more favorable in stable environmental conditions. And that's because less energy and resources are needed for it to occur. And genetic diversity is not as important, or increasing the current genetic diversity is not important. And this is because the organisms that are currently living in this environment are surviving just fine with the genetics that they have. Sexual reproduction, however, is typically more favorable in a changing or unstable environment. And this is because sexual reproduction can increase genetic diversity, which can help the population to survive if conditions are changing.